Hey guys, welcome to Medifaction. Today, let's learn about the physiology of cell junction. Cell junctions are a class of cellular structures consisting of multi-protein complexes that provide contact or adhesion between neighboring cells or between a cell and the extracellular matrix in animals. So, in order to learn more about cell junctions, let's get started. In this video, I will be enumerating an introduction, types of cell junctions which are occluding junction, communicating junction and anchoring junctions. We will be also talking about adherence junction, focal adhesion, desmosome, hemidesmosome and some applied physiology related to cell junctions. Introduction. Cell junction is the connection between the neighboring cells or the contact between the cell and the extracellular matrix. It is also called membrane junction. Cell junctions are classified into three types. Number 1, occluding junctions. Number 2, communicating junctions. Number 3, anchoring junctions. Occluding junctions. Cell junctions which prevent intercellular exchange of substances are called occluding junctions. That is, these junctions prevent the movement of ions and molecules from one cell to another cell. Tight junctions belong to this category. Therefore, let's now learn about tight junction. Tight junction is the intercellular occluding junction that prevents the passage of large molecules. It is also called zonular occludence. It is the region where the cell membranes of the adjacent cells fuse together firmly. This type of junction is present in the apical margins of epithelial and endothelial cells, in the intestinal mucosa, wall of the renal tube, capillary wall and also choroid plexus. Structure of tight junction. Tight junction is made up of a ridge which has two halves. One half of the ridge is from one cell and another half of the ridge is from another cell. Both halves of the ridge fuse with each other very tightly and occupy the space between the two cells. Each half of the ridge consists of tight junction strands. Function of tight junction. Number 1 is strength and stability. The tight junction holds the neighboring cells of the tissues firmly and thus provides strength and stability to the tissues. Next function is that selective permeability, otherwise gate function. The tight junction forms a selective barrier for small molecules and a total barrier for large molecules. In the epithelial and endothelial cells, tight junction is the most apical intercellular junction which functions as selective, that is, semi-permeable diffusion barriers between the neighboring cells. This function is called barrier or gate function. Barrier function of tight junction regulates the interchange of ions, water and varieties of macromolecules between the cells. The magnitude of this function varies in different tissues. In some epithelial tissues or cells, few substances pass through the tight junction. That will be by diffusion or active transport. In other cells, no substance passes through the tight junction. Third function, which is fencing function. Tight junction prevents the lateral movement of proteins, that is, integral membrane proteins, and also lipids in cell membrane, and thus it acts as a fence. The fencing function maintains the different composition of proteins and lipids between the apical and basolateral plasma membrane domains. Because of this function, the tight junction is sometimes also referred as impermeable junction. Fourth function, maintenance of cell polarity. Fencing function of the tight junction maintains the cell polarity by keeping the proteins in the apical region of the cell membrane. Fifth function, blood-brain barrier. Tight junction in the brain capillaries forms the blood-brain barrier, which prevents the entrance of many substances from capillary blood into brain tissues. Only lipid-soluble substances like drugs and steroid hormones can pass through the blood-brain barrier. Now let's learn about communicating junctions. Cell junctions, which permit the intercellular exchange of substances, are called communicating junctions. That is, these junctions permit the movement of ions and molecules from one cell to another cell. Gap junction and chemical synapse are the communicating junctions. So let's learn about gap junction. Gap junction is the intercellular junction that allows passage of ions and smaller molecules between the cells. It is also called nexus. It is present in heart, basal part of epithelial cells, of intestinal mucosa, etc. Structure of gap junction. Membranes of the two adjacent cells lie very close to each other, and the intercellular space is reduced from the usual size of 2.5 to 3 nanometer. Cytoplasm of the two cells is connected by the channels formed by the membranes of both cells. Here you can observe that cytoplasm is fused. So the molecules move from one cell to the another cell directly through these channels without having contact with extracellular fluid. Each channel consists of two halves. Each half belongs to one of the two adjacent cells. Each half of the channel is surrounded by 6 subunits of proteins which are called connexins or connexons. So that is about structure of gap junction. What is the function of gap junction? Number 1, diameter of the channel in the gap junction is about 1.5 to 3 nanometers. So the channel permits passage of glucose, amino acids, ions, and other substances which have a molecular weight less than 1000. Second function, it helps in the exchange of chemical messengers between the cells. Third function, it also helps in rapid propagation of action potential from one cell to another cells. Third type, that is anchoring junctions. Anchoring junctions are the junctions which provide strength to the cells by acting like mechanical attachments. That is, these junctions provide firm structural attachment between two cells or between a cell and the extracellular matrix. Anchoring junctions are responsible for the structural integrity of the tissue and are present in the tissues like heart muscles and epidermis of the skin, which are subjected to severe mechanical stress. The firm attachment between two cells or between a cell and the extracellular matrix is provided by either actin filaments or the intermediate filaments. Depending upon this, anchoring junctions are classified into 4 types. Number 1, actin filament attachment. It is subdivided to adherence junction, which is from cell to cell, and also focal adhesion, which is from cell to matrix. Next type is intermediate filament attachment. This is again subdivided into desmosome, which is cell-to-cell connection, and hemidesmosome, which is the connection between cell to matrix. Adherence junction. Adherence junction is the cell-to-cell connection or cell-to-cell junction which connects the actin filament of one cell to those of another cell. Here you can appreciate that. In some places like epithelial linings, this junction forms a continuous adhesion, that is zonular adherence, just below the tight junctions. In adherence junction, the membranes of the adjacent cells are held together by some transmembrane proteins called cadherens. Adherence junction provides strong mechanical attachment of the adjacent cells. Adherence junction is present in the intercalated discs between the branches of cardiac muscles. During the contraction and relaxation of the heart, the cardiac muscle fibers are held together tightly by the means of this junction. The adherence junction present in epidermis helps in the skin to withstand the mechanical stress also. Focal adhesion. Focal adhesion is the cell to matrix junctions which connects the actin filaments of the cell to the extracellular matrix. In epithelia of various organs, this junction connects the cells with their basal lamina. The transmembrane proteins which hold the cell membrane and the matrix are called integrins. Next one is desmosome. Desmosome is a cell-to-cell junction where the intermediate filaments connect two adjacent cells. Desmosome is also called macular adherens. The membranes of two adjacent cells which oppose each other are thickened or become spot-like patches. Intermediate filaments are attached with the thickened patches. Some of these filaments are parallel to the membrane and others are arranged in radiating fashion. Desmosome functions like tight junctions itself. The transmembrane protein involved in desmosome are mainly cadherens. Next is hemidesmosome. Hemidesmosome is a cell to matrix connection or junction which connects the intermediate filaments of the cell to the extracellular matrix. This type of cell junction is like half desmosome and the thickening of the membrane of only one cell occurs here. So this is known as hemidesmosome or half desmosome. Mostly the hemidesmosome connects the cells with their basal lamina. The protein involved in this are integrins. Now let's learn about some important applied physiology. Number 1. Dysfunction of adherence junction and focal junction in colon due to mutation of proteins results in colon cancer. It also leads to tumor metastasis, which means spread of cancer cells from a primary tumor to other parts of the body. Number 2. Dysfunction of desmosome causes bullous pemphigoid, which is an autoimmune disease with tense blister formation in the skin, like here. The patient with this disease develop antibodies against cadherens. Number 3. Dysfunction of hemidesmosome also cause bullous pemphigoid. The patients develop antibodies against integrins in this condition. Hope you have understood the video. Like, subscribe and press the bell button for more videos. Thank you. Thanks for watching.